Good morning, class. Welcome to the second session of biology, class 10th. In the last session, we had started the first chapter, that was cell cycle and cell division. Let's have a recap of the topics that we had completed in the last session. In the last session, I told you about cell cycle. What is cell cycle? I had given you the definition and a deep explanation about cell cycle. I told you the meaning of generation time. We also discussed various phases of cell cycle that included the interphase, that is the non-dividing phase. It included the G1 phase, S phase and G2 phase. I had told you the full form of all these three, where G stands for gap or growth, S stands for synthesis or synthetic phase. In the same way, G2 also gap and growth phase. And I had told you for different species, the time taken for the cell division is different. That is, for the time of that interphase will differ. I had told you the phases of cell cycle, that was the interphase, was a non-dividing phase, and then came the actual division phase, that was division phase that included the karyokinesis and cytokinesis. Karyokinesis, I had told you, it is the division of nucleus. Then, and cytokinesis is the division of cytoplasm. The types of cell division, I had told you, there are two types of cell division that occur at various places of the living cell body, of the living body. The first one is mitosis. Mitosis occurs in two phases. The first one is karyokinesis, that is the division of nucleus, and the second one is cytokinesis that I had told you. This karyokinesis, when we are talking about, it is also called as the nuclear division. Why it is called as the nuclear division? Because in this phase, the nucleus divides. The various phases that I had told you in the class that constituted the karyokinesis were prophase, metaphase, anaphase, telophase. I had explained you all these phases with the help of diagrams. Then coming to cytokinesis. Cytokinesis is the process where the actual division or the division of the cytoplasm takes place. But this division of cytoplasm or cytokinesis differs in animals as compared to that of the plants. In animal cells, the cytoplasm divides by constriction. I had told you, by the formation of a furrow at the place of equator. It begins on the equator and it gradually starts to deepen inside and converges from the sides. Whereas in the plant cells, I had told you, the cytoplasmic division or the cytokinesis takes place by the formation of cell plates. Cell plates are formed. These cell plates which are formed are formed by the fusion of vesicles, and these vesicles are produced by dictyosomes. What are dictyosomes? These are the Golgi apparatus. They are called as by the name Golgi apparatus in animal cell. In plant cell, they are the dictyosomes. Begins in the middle. What actually happens? Formation of the cell plate will start in the middle and it will start to move towards the periphery. As it will move towards the periphery, it will become continuous, and from that point itself, the two daughter cells will get detached. Now, we will be going to start today's session. Today, first of all, we will be understanding what are the significance of mitosis. Importance of mitosis. The very first importance of mitosis is growth. We all know growth cannot be possible without mitosis. So mitosis is very much important. You can see growth, then replacement. As I had told you, as the older cells complete their life cycle, they need to after wear and tear, they are shed off. So that place is taken by new cells, and that is replacement. Then comes size. The size of the cell the size of the cell depends, it also depends on mitosis, means a cell will grow, isn't it, by this mitosis. It helps, the mitosis helps in maintaining its proper size. Then repair, I had told you, due to some cuts or wound, whenever we get a cut or wound, the repair process is brought about by mitosis. Then reproduction, basically when we talk about reproduction in unicellular organisms, here the reproduction takes, or asexual method, when the reproduction is taking place, it is done by this process of mitosis. So this was the significance where we had seen growth, replacement, size, repair and reproduction. Now we will see one very important thing which is related to mitosis, that is cancer. Cancer is the result of uncontrolled mitosis. I had told you about what is mitosis, what actually happens in cancer. You might have heard this disease, the name very commonly you might have heard about cancer. So what actually happens in cancer, thus the cells, they undergo they go cell division. First of all the karyokinesis and then followed by the cytokinesis. This all division is controlled. When it becomes uncontrolled, what happens, it develops masses of cells, and these masses of cells are called as tumor. This mass of undifferentiated cells without any shape and size, they are called as tumors. Cells divide by uncontrolled way. Why do they, what happens that they start to divide in an uncontrolled way? It happens sometimes due to some genetic changes, that is called as gene mutation, or sometimes due to the presence of some oncogenes. These oncogenes, they destroy or they disable the checkpoints that prevents the cell division. Actually there are some checkpoints that they will regulate the rate, or the speed of cell division. So these checkpoints are disabled, or sometimes they are destroyed by these oncogenes. Now let us see the next point, that is difference between mitosis. I have told you that mitosis occurs in animals as well as in the plant cell. So what is the difference, the basic difference between the mitosis of animal and plant cell? So talking about the karyokinesis, it will be somewhat same. That is, the nuclear division will be same in animals as well that in the plant cell. The difference will come at the time of cytokinesis. If we see the first difference between animal cell and plant cell is this: an animal cell will possess a centriole. Centriole is a very important organelle which is found in the animal cell, whereas it is absent in the plant cell. Actually, this centriole initiates the process of cell division in animal cell. How does it initiate? As it divides, this centriole divides into two and forms ester. Okay, and from that only estral rays arise, which forms the spindle fiber, which further continue the division of the cells. So the first difference between this animal and plant cell mitosis is that animal cells, they possess a centriole, whereas the centriole is absent in plant cell. Secondly, when centrioles are present in the animal cell, they will form ester. Ester formation takes place from the centriole. As you all know, that I had told you in the previous class, the centriole divides into two. It moves towards its respective poles. The estral rays are formed. The estral rays then it arises from there the spindle fibers. So whereas since centrioles are not there in plant cell, ester formation does not take place. In animal cell, at the time of cytokinesis, it occurs due to constriction. I told you a constriction develops in the equatorial portion, whereas in the case of plant cell, mitosis occurs by the formation of cell plate. Then cell division in tissue throughout the body. Very important difference between the one very important difference between the mitosis of animal cell and plant cell is that mitosis will in animal cell, it will take place in all in the cells, in the tissues of all throughout the body, whereas in the plant cells that it is only localized to some particular tissues, or the tissues or the cells which are found in the growing region, which basically constitute the meristematic cells. So these are the 4 points or the 4 differences between animal cell and plant cell. Now we will be starting with meiosis. What is meiosis? Meiosis is also called as the reduction division. Why it is called as reduction division? The reason, the answer is they are called as reduction division because the number of chromosomes is halved. Actually, what happens? 4 haploid daughter cells are formed during meiosis from a single diploid parent cell. A single diploid parent cell is there. I have told you the meaning of diploid and haploid cells. Diploid cells are those where homologous pair of chromosomes are present, whereas in haploid only one chromosome is present. So in meiosis, 4 haploid daughter cells are formed from a single diploid, that is to an parent cell. I told you chromosome number is halved. I'll tell you why it is halved and where does it occurs. Where does this where will this meiosis where will it occur? It will occur in all the sexually reproducing organisms, basically in their sexual organs, and it is necessary at the time of formation of gametes. Now, what is the need of meiosis? Meiosis, the basic need of meiosis is to maintain the number of chromosomes. As we all know, suppose any species, if we talk about, suppose human beings, if we say they are having 46 chromosomes, okay, 23 pairs each. Then suppose the 2 cells are fusing. Okay, suppose the gametes are diploid. What will happen? The offspring or the zygote will, the number of chromosome will keep on increasing. In order to maintain the number of chromosome fixed in a particular race, the number of chromosomes is halved at the time of formation of gametes. To maintain the chromosomal number of a species from generation to generation, we all know all the individuals of a particular species, they will have a particular number of chromosomes. How it is possible? It is possible due to this meiotic division. All body cells are diploid. We all know our body cells all together, they are diploid. When a zygote is formed by the, we know when the zygote is formed, we know it is formed by the union of male and the female gametes, that is the sperm cells and the egg cells. If the gametes will be diploid, then every time during fusion the chromosomal number keeps on increasing, due to which the number of chromosome will not be stable, that I have already told you. Therefore, chromosome is halved during gamete formation. Now, what actually happens when if we see the structure of chromosome, we can see here the structure of chromosome which is given here. This is a chromosome, okay, a single you can see chromosome which is having a centromere, which is the point of attachment, and 2 arms. These are the arms of the chromosome. This is the structure of the chromosome. Upon its duplication, what will form? It forms 2 sister chromatids. 2 sister chromatids are also attached to the centromere itself. What happens during gamete formation? We can see here, I have shown you here paternal chromosome and maternal chromosome in this way. Okay, it is present in the gametes. So paternal chromosome, you can see here the arms of paternal chromosome, and maternal chromosome, you can see here the arms of the maternal chromosome. Okay, upon its duplication, you can see here 2 sister chromatids are formed in the maternal chromosome side as well as in the paternal chromosome side. Whereas you can see, that is why I have made it with different colors also, okay, to differentiate it. Now, The non-sister chromatid, you can see the the chromatid, the chromosome of this maternal side and the chromosome from the paternal side, they are called as the non-sister chromatids. Okay. Now, when we see the process of meiosis, it undergoes two subsequent divisions. When we are coming to the process of meiosis, we will see a PowerPoint presentation first of all. We will see PowerPoint presentation. The kind of cell division that produces the sex cells and gametes. It takes place in the reproductive organs, that is testis and ovary, anthers, and the ovary in the plants. The most significant aspect of meiosis is that the number of chromosomes in sex cells is halved. For example, human beings have 46 chromosomes, that is 23 homologous pairs. The two similar chromosomes of diploid cell, that is 2n, is called homologous pair. The chromosomes of homologous pair are brought together by the zygote, by the union of the male sperm, that is sperm, and in female the egg cell gametes. Therefore, one chromosome in the homologous pair is maternal and the other one is paternal. When two haploid gametes, that is sperm and egg cell, fuse, the resulting zygote has a full diploid set of chromosomes, one set of each kind. In this way, the chromosome number is maintained constant from one generation to the other. Then process of meiosis, the cell undergoes two subsequent divisions, that is the first reduction division, meiosis 1. Second division is a simple mitotic division, that is meiosis 2. In meiosis 1, the chromosome number is reduced to half as the chromosome from each pair gets separated and distributed equally in two cells. During division, there may be exchange of chromatids, segments of homologous chromosomes. This is what called as crossing over. This exchange of chromatid segments is called as crossing over. Then crossing over results into exchange of genes and produces new gene combination in the offsprings. In meiosis 2, chromosomes of daughter cells break up into two chromatids and daughter cells undergo further division containing one chromatid of each kind. As a result of haploid daughter cells, 4 haploid daughter cells are formed. What is the significance of this meiosis? The basic significance we will be seeing one by one. The first one is meiosis maintains the number of chromosomes in a species generation after generation. Second one, meiosis causes mixing of maternal and paternal chromosomes in the gamete. Third one, meiosis results in recombination of new combination of genes. Fourth one, meiosis introduces variation among individuals. So you had seen the PowerPoint presentation. Now the process of meiosis, as I told you, it undergoes two subsequent division. The first division is the reductional division, that is meiosis. Why it is called as reduction division? Because the number of chromosome is made half. And the second one is meiosis second, or which is a simple meiotic division. Let us start to understand the process of meiosis. If we can see here, this is the structure of the nucleus, where we can find the chromatin fibers scattered or in the nucleoplasm. You can see here the point of attachment, that is the centromere present in the chromatin fibers. What actually first of all happens? So what kind of cell is this? It's a cell or a nucleus which is diploid. What actually happens then? The duplication of the chromosome first of all will take place. If we can see here, I have shown again the chromosomes with two different colors, which denotes that this blue one, the paternal side, and the red one, the maternal side. Okay. So what actually happens? The homologous chromosome, you can speak. First of all, we will see chromatin fibers. They start to condense. After condensation, they will form this duplicated form of chromosomes. Okay, you can see here, and homologous pairing will take place. What is homologous pairing? The homologous pairing means chromosomes of homologous pair means of same size and shape. So those chromosomes which are of same size and shape are termed as homologous chromosomes. So homologous pairing, same type of chromosome which is coming from the maternal side and the paternal side, we will find them coming together, which is called as homologous pairing. We will find here, we can see a homologous pairing is taking place. Now after this homologous pairing, what will happen? Crossing over will take place. Now one thing you have to be very clear, that crossing over, it is not sure that it will take place in all the homologous pair. It may take place. In some it may take place. So suppose in this one it is taking place, the crossing over is taking place. What is crossing over? Overlapping of the chromatids. You can see here overlapping of the non-sister chromatids. It will take place, and the point of attachment, or the point where it is overlapping, it is called as chiasmata. Okay, and from here, from this point itself, the segments will break and recombination of genes will take place. We can see here the colors as they are being exchanged. Here we will find recombination of genes has taken place due to crossing over, whereas in this one where there is no recombination taking place. That is why I had said that it is not that it will take place in all the combinations. This after division, what will happen? After homologous pairing and exchange of chromosomes will take place. This is called crossing over. What will happen? Exchange of segments will take place. That will result in the mixing of genes in the generation. It will result into the new combination of genes. What will happen then? The separation of that, you can see here two cells are formed with combination of genes from there. Each one combination they are getting. Okay, then these two will again get divided into the gamete, that is haploid gametes will be formed, and each one will have only one segment of the chromosome. We can see here, from here it breaks, half it comes here, half comes here, from here also it divides into two. This one also it will break and haploid gametes are formed. What is the significance? I had told you in the PowerPoint presentation also that due to this meiosis or the meiotic division, the chromosomal number is maintained. It does not increases. Mixing of maternal and paternal chromosomes. What happens? Homologous pairing that takes place, and due to the mixing, what happens? Recombination of genes takes place, and introduction new variations are introduced. among the individuals. So we can see variations are introduced due to this meiosis. Difference between mitosis and meiosis. Now we'll be seeing the difference between, what is the difference. I had told you, I had explained you about mitosis, and I have also explained you about meiosis, that is the reductional division. So in mitosis, the basic difference that we are going to see in mitosis and meiosis is that mitosis will take place in all the somatic cells, that is the body cells, whereas I had told you earlier also, meiosis will take place in all the reproductive cells, that is during the formation of the gametes. Then mitosis, in mitosis it is a kind of division which is also called as equational division, where the number of chromosomes is same as that of the parent cell, whereas in the case of meiosis it is a reductional division where the number of chromosome is halved. Then in mitosis two similar cells are formed, and both the cells are diploid, means two N chromosomes are present, whereas in meiosis four haploid gametes are formed, which are called four haploid cells or gametes are formed, which is represented by N. So this was all about today's topic. We have also completed the chapter, and I'll be giving you some questions in regard to the chapter. We'll be seeing some of the questions, like name the types of cell division, why is interphase named resting phase, what is karyokinesis, why is first meiotic division called reduction division. Let us see. So please note down these homeworks, and you can write this the answer of this question in any copy. You can also use the previous 9th class biology copy to write the answer or the homework. You can use that, or you can use any other revision copy, or any you can make a rough copy, or a new copy you can make. Okay, so let us see what is the homework questions. Name the types of cell divisions. Why is interphase named resting phase? What is karyokinesis? Why is first meiotic division called reduction division? Please note down these questions, and you can write the answer. All for today's lecture. Goodbye.